And now, to refute the previous lecture on Mendelian genetics, here's non-Mendelian genetics. Okay, not so much refute as add on to. In this lecture, please define incomplete dominance, carrier, multiple alleles, codominance, polygenic inheritance, linkage, crossing over, intersex, sex linkage, and sex-influenced genes. Be able to compare methods of inheritance, relate inheritance to the ABO blood types. That's been coming up a lot, hasn't it? Relate intersex and other variations at birth to current legislation. That's more for people who are in social work and nursing. And be able to draw Punnett squares and pedigrees for each inheritance pattern. That's a great way of studying. Incomplete dominance. This is where we have a dominant phenotype, a recessive phenotype, and a heterozygous or intermediate phenotype. A Punnett square will give a 1 to 2 to 1 ratio not only of genotypes, but of phenotypes. And this means the heterozygote is going to be the intermediate type. One example of this will be the sickle cell anemia, where we have incomplete dominance. We can have individuals who are fully sickle-celled if they have both recessive alleles. We have individuals that are normal if they have both normal alleles. And we have heterozygotes, which have some sickle cells, but not all sickle cells. This actually gives an advantage in the presence of malaria because the malaria pathogen cannot invade sickle-celled blood cells, but can invade normal blood cells. So the sickle-celled blood cell individuals that are completely sickle cells have a disadvantage because it's sickle cell anemia, which is deadly. The normal blood cell individuals have a disadvantage because they have, susceptible to malaria cells, and the intermediates actually have an advantage over both. And this is where we get the idea of a carrier as well. It's someone who has the recessive allele but doesn't express it. And honestly, in the incomplete dominance, they do express it somewhat. So codominance here means that we are going to be able to express more than one alleles at the same time. So you get both the A and B blood type alleles, and they are presented in AB blood. It's also notable that both of these are dominant to O in a simple Mendelian manner, and the Rh factor, the plus or minus, is inherited in a strictly Mendelian factor. So the ABO blood types are both codominant between A and B, and both dominant to O. So let's do some crosses here. We have your A blood type shown by the IA and the B blood type shown by the IB. You have two AB individuals, and this is what they produce. One type A, two type ABs, and one type B. So we're going to calculate the likelihood of each. And there's your type B, even though they had a type, if you took them and both made them with a type A partner that had type A, but also O, as you see, you can get even more blood types, but you cannot get O unless there are two O alleles, one on each parent. So polygenic inheritance is another thing we're going to look at here, and that's where they have more than two genes for a trait. Something like height is polygenic. There isn't simply a short and tall human. We all have a range of heights. We also have intelligence is polygenic. There isn't a brain gene and a lack of brain gene. It's rather polygenic and exists along a gradient. Skin color is polygenic, and in a purely mixed population, we'd actually see mostly intermediate phenotypes. Now, what we see here is we see a gradient, and when we see a gradient, we can infer that this is polygenic. Likewise, you might think skin color, wait, isn't it November when I'm watching this? Didn't he film this in August? Didn't his skin color change? Well, we're also talking about inheritance and environment, and these are two different factors. Environment can change things a little bit. Environment changes height. If you're well fed when you're young, you get taller. If you're well educated when you're young, you get smarter. If you're out in the sun a lot and your skin can respond to that, you become darker. So there is a certain amount of environmental factors that play into polygenic inheritance, and the way they do so is by turning on or off or accentuating certain genes. We also have something called heterosis, which if you were to take two of those individuals that were at the middle of that chart right there showing different skin pigments, and they were both the same skin pigment, but because of different alleles, and they had offspring, you could hypothetically get someone far darker or far lighter, and that's called heterosis, when two individuals who are alike produce offspring that are not alike, either of them. And this does mean that two very intelligent individuals can have a kid who's not very intelligent, or two medium intelligent individuals can have two children, one of whom is very bright and the other of whom, well, maybe they've got a job in Congress. Polygenic inheritance is also related to human migrations. Humans originated in the Adar Triangle of Africa, and if we look at where that is, it's a spot where there is much darker skin color. That was the original humans. They had much darker skin, and what we do see is actually those alleles being selected against when humans moved into places like Northern Europe. And there are two possible reasons for this that we're not getting into right now. We can also use polygenic inheritance to measure things like the Bantu expansion in Africa, where we can actually look at the movement of certain alleles through Africa as the Bantu Empire expanded, or the movements of Vikings in Europe. They actually had distinct alleles, and we can follow these alleles from the Vikings in Finland and Norway and Sweden. We can follow them into England and even as far as Malta. And we can also follow humans as they traveled across the Bering Land Bridge to arrive in North America. And this is useful when we're looking at polygenic inheritance and how the environment has factored in on this. You can see that individuals in South Africa do not have as dark of skin as those individuals in Africa. And one of the reasons for this is they lost certain alleles as they migrated. Genetic linkage. Well, this just violates the law of independent assortment all over. Didn't we say that different alleles for different genes are going to be inherited independently and assort independently? Wah! Well, for the most part, yes. But if they're right next to each other, if they're very linked, if they're very close to each other on the same chromosome, they'll assort together during metaphase 1. But they can cross over during prophase. The likelihood that they will cross over is related to how close those different genes are on a chromosome. We're not going to get into this a lot. I welcome your questions. I welcome your questions, and Dr. Olney's really good at this, but we're not really covering a lot of this in class. Maybe in a review session, maybe just for fun, but not in class. Sex determination. So there are genes that are sex determined, and here we run into a wonderful thing about our modern society. So the default of a human when they're being built in the womb is female. The presence of the Y gene, Y chromosome, and specifically a certain gene called SRY begins making someone male. In fact, if an individual has an X chromosome but nothing else, just a single X chromosome, they turn out looking entirely female. If an individual has a Y chromosome and no X chromosome, they're dead. They can't function, so the embryo does not live. So what this tells is the default, well, it's female. And when we have some things like this in society where we've got your gender mixed up with sex. Sex is a biological construct. It's that male and female, that sperm donor and egg donor I've been mentioning. Gender is a societal construct, and it's notable that not all societies have genders. Some societies just have sexes, and that's what's different in different languages. We also have very odd societal constructs like alpha male, which is kind of pseudoscience for now, but might eventually become mainstream science if certain factions have their way. We also have non-binary, which is a gender, but that's a societal construct. If you asked someone in a language where they didn't have genders, if they identified as non-binary, they wouldn't understand what you're talking about. So misconstruing sex and gender is a way to make the argument simpler, but a lot dumber. There are also pro-male and anti-male genes. So the SRY gene actually activates other genes. And these other genes are your sex-specific genes. What this means is we have polygenic inheritance. Remember, polygenic inheritance creates somewhat of a gradient. So there might be different phenotypes as opposed to the strict XY is male, XX is female. There's actually a lot more going on there. And specifically, here's your intersex and other complications. You have your normal XY male that has the SRY gene on the Y chromosome. If during prophase 1, you have recombination between the Y chromosome and the X chromosome, which normally you do not, but sometimes you do, you can end up with the SRY gene going over to the X chromosome. And if that person has a daughter, you'd have XX. And did I say daughter? I meant son, because they have the SRY gene, and that makes them a male with 2 X chromosomes. That same individual, having had recombination on the Y chromosome, could also give rise to a son with XY. Did I say a son? I meant a daughter, because that would be female with an X and a Y chromosome. There's also intersex, which can be intermediate sex forms. And if you remember the reproduction slide, well, we can talk more about the reproduction slide in class, but some of the things that were taught on that are very not appropriate for certain states. You can also have androgen exposure in utero, which means if the mother is taking testosterone or testosterone supplements, what will happen is that will actually turn on some of those pro-male genes, and that will result in a more male phenotype. We can discuss the specifics of this in class, but again, this is YouTube. Sex linkage. This is where we have something that is actually on the X chromosome. The X chromosome has way more genes than the Y chromosome, in the same way that I have more books in my library than I have in my office. It's a lot more genes. Males may be monozygous for genes in the X chromosome because that just means they have, well, one copy. They can't be homozygous or heterozygous if you only have one copy. Examples of this are color blindness, your red-green color blindness, and hemophilia. What that means is a mother would not have hemophilia. She would be a carrier for the hemophilia gene. And the father would not have hemophilia because he doesn't have the hemophilia allele. But he can have a daughter. They can have a daughter that's a carrier and a son with hemophilia based on the sex linkage of this X chromosome-based gene. And here is a pedigree of such where you see individuals who are carriers, they've got the little dots, and individuals who have hemophilia, those are the filled-in blocks. Because there are 2 X chromosomes, sometimes you have one of those X chromosomes just condensed and is inactivated, so the X chromosome doesn't really get read, allowing only one X chromosome to be expressed. So here we have a tortoiseshell cat, and those tortoiseshell cats have different alleles, one for brown skin color, well, hair color, and one for black hair color, and those are expressed on the different X chromosomes. But since at random, one X chromosome is going to be inactivated, you end up with this mosaic organism, which, really pretty, but will probably claw your arm off.